Good morning. We're going to get underway. If I can get everybody's attention, we're going live over the internet starting right at 9 a.m. So I'm going to need to have, unless you forged the signature from Alan Crow, which maybe half of you did. Anyway, all right, so a couple other announcements here. The silent auction is going to close at 12 o'clock. If you want to get your last bids in, make sure to have them in before that. The evaluations, I already told you, are important, and we really need your feedback, and we do incorporate those into planning for next year, so do not forget to return those. Last thing is, everybody in this room, be aware that we're live over the internet. The cameras are only going to focus on the speakers up here. The other videotaping we've been doing as well, I want you to understand that we do not shoot the faces of people here at the conference. So if you're concerned at all about your identity, privacy, it has been protected. I'm going to stop there and make sure we have everybody. Okay. All right. I want to introduce to you our Research Director and Communications Director, Linda Cobert. She's going to moderate this panel. The one thing I want to let you know, we're going to let all of our medical advisors have an opportunity to talk about themselves and their work. We're going to save questions till afterward. We may have some questions, and not too many, after the very first presenter because he needs to leave, and it's also on a clinical trial that I know is of interest to many of you. So let me turn it over to Linda.
Thanks, Bob. How's everybody doing? Great, great. So as Bob said, we have a panel of 22 of the most expert people in myositis research with us today, and we're going to hear from each one of them. But first, we're going to start off with Dr. Domachky. Many of you are really interested in finding out about the arimoclomol clinical trial that is starting to recruit, and we thought we would get the principal investigator to give you the updates as we know it. So we will spend 5 minutes to have him give a little talk, and if there is time, we'll take a question or two. But please be reminded that the drug company Orphazyme is here exhibiting, so you can also go to them to get their literature and talk to them. Dr. Domachky?
Yeah, thanks, Linda, for the introduction. I'm Mazen Domachky. I'm guilty as charged, meaning that I'm the principal investigator for this international study of arimoclomol in inclusion body myositis. Can everybody hear me well in the back? Yes? Okay, great. So we're really at an exciting time of this study. It's really been gratifying to see that our efforts to get the sites around the United States activated is moving forward at a much brisker rate than I would have anticipated, meaning that my predictions based on the current behind-the-scenes activities that I'm seeing throughout the course of the study is that the sites in the United States will be activated by October of this year. As you know, we have 11 sites in the US. 2 of them are active right now, University of Kansas and the Nerve and Muscle Center in Houston, Texas. There'll be sites on the East Coast, West Coast, South, center of the US, dispersed throughout the United States. And so we're excited that we're getting closer to that time point in the activation of the United States sites. University of College London has already screened a patient, and so we're really excited about that too. So it's a large study. It's about 150 patients, volunteers we're looking for. So thank you for your patience, but also for your sustained interest in playing a big role in this study. So we really appreciate you and your interest in that. And I apologize profusely for having imposed so much on your patients, but all our efforts behind the scenes have been for the common cause. The common cause is finding out, number one, finding out if this drug works for IBM, and number two, it would be a total waste of your time and our time, but more importantly, your time, if we did the study, didn't do the right setup for it, for if the study is showing the drug to be working, for the FDA to look at the data and say, it's not good enough, you have to do another study. That would be very frustrating. So a lot of our efforts behind the scenes have been to guarantee that if the study is positive, that the FDA would look at the data and said it was done in a good way. So thanks everybody for your patience, and I apologize for any frustration this wait has caused for you. Thank you.
I think we might have time for one question. Is there somebody? Do we have a microphone that we can bring up here? Sure.
Yeah, so from what I understand, because I always thought the selections of patients was already finalized at all the different U.S. locations. I'm looking at the University of Virginia, I believe, is doing one. And Duke has submitted me to be part of that study, but I haven't heard anything. So is it still in process of recruiting?
So this is a very good question. It just reflects the general structure of how research is done. For example, you mentioned University of Virginia. I'm going to tell you my response. I cannot confirm or deny if the University of Virginia is a site or not. Okay? So that's number one. Number two, the process, and then some of the things you probably are asking me, or the undertone of the question is the following. Well, what can I do now while I'm waiting for some site closer up to me to open up? Because that's, you want to do something, you want to be ready. You don't want to just get to the opening of a site and then say, oh, but you need to do this. So my advice to interested patients is to gather your notes from your neurologist, from your rheumatologist, and have your muscle biopsy reports available so that when your number is called to come to that site that's closest up to you, then you'll be able to just have everything ready to go. Now, the next question will be, well, how will I know that my site closest up to me is going to be active, right? And we're updating this continually on clinicaltrials.gov. If you look up clinicaltrials.gov, and then there's a search term, and you put in it arimoclomol, only 4 or 5 studies will show up. One of them will be the IBM study. You click on it. At the bottom of that is the list of active sites. So if any of you logged in right now, you will see 3 active sites: University of College London, University of Kansas, Nerve and Muscle Center in Houston. And as more sites get activated, this will be updated. Why do I know this? Because these 2 sites were activated in the last 2 weeks. We're that quick in updating the site, because we want people to have access to that information, beyond which there's also an email link to my research program manager for the study, Tina Liu, who can update you if you have a question about whether your site close up to you is or not active. Does that answer your questions? Thank you.
Thank you, Dr. Dimachky. So let's start introducing our panel, and by way of introduction, I thought it would be a good idea to tell some stories. So as we all have asked patients to tell their myositis journey, the story of their myositis journey, I'm going to invite our panelists to tell that story about themselves. And we'll alternate sides. We'll start over here with Dr. Vlugels.
Well, since I've been put on the spot, here is my myositis story. I became very interested in dermatomyositis when I was actually in training at Harvard as a resident, when I met my first patient with this disease. And I decided at that time that I really wanted to become dedicated to studying and seeing patients with this condition. I am the dermatologist on the panel here. And I see both adults and children. So I work at Brigham and Women's Hospital and Boston Children's Hospital. And I follow now several hundred patients with this disease. And sort of the most recent thing I've been doing is working with one of my neuromuscular colleagues to help design a drug that's used specifically to treat patients with cutaneous dermatomyositis, or skin disease and dermatomyositis, excuse me, that's now in a trial at 12 sites in the United States, and that's been really fun for me to work on. But really, I just love seeing and taking care of patients with this condition and really trying to get patients doing as great as they possibly can. Thanks.
So next, I'm sorry, I can't see, I think, Dr. Schmidt.
So, hello everybody, my name is Jens Schmidt from Göttingen in Germany. I'm a neurologist by training, and I'm head of the Neuromuscular Center in Göttingen, so we see patients with all sorts of neuromuscular diseases, neuropathies, myopathies, and of course, myositis. So my special interest in myositis started during my fellowship here in the US at the NIH in Bethesda. That's a couple of years ago, and I started my own group in Germany. We have a strong basic science and translational science program. And over the years, having contact to many patients with myositis, I saw that there was a need for the patients to come together, and I was a co-founder of a German patient support group for myositis. And over the years, I stepped back, and now I'm a medical advisor for that patient support group in Germany. And I decided to still become active overseas and reach out to the TMA, and here I am.
Thank you. And Dr. Reed?
Good morning. My name is Ann Reed. I am an immunologist and pediatric rheumatologist by training. I presently am at Duke University, but my interest in myositis began when I was training and got exposed to children who had myositis. At that point in time, seems like many years ago, because it was, really started getting interest in understanding the genetics behind the disease and the predisposition for the disease. And I actually did a lot of early work trying to help us define that and understand what it means. And then my work evolved and my interest evolved as I think technology evolved, and we were able to then refine further sort of the biology of myositis, and particularly dermatomyositis in children for me. And more recently have evolved my work into looking at markers of disease activity to try and help improve our ability to predict when an individual is improving in their condition, worsening in their condition, and really to try to predict some treatment outcomes. And so I've expanded some of that into adult myositis as well, but still focused primarily on dermatomyositis.
Thank you. Sue Millard?
Good morning. I'm Sue Maylard. I'm actually a specialist physiotherapist from London, and I work with young people with myositis. My interest started very young into my career when I met a young man who had myositis, and I was instructed that we couldn't exercise him because he had active disease. And 9 months went by, and by the time I was allowed to work with him, he had no movement, no strength, and had almost no life. And though it took me a year, and I gave, together we got back his mobility and his strength, and he was able to leave hospital and go back to school, and his family, it made me think that that can't possibly be the correct way of managing myositis. So with my institution, we looked at whether exercise was safe from the diagnosis of myositis rather than waiting for a medical approval that the disease was inactive. And we therefore have changed completely the way that we manage myositis in young people. And now if a young person is diagnosed with myositis, they start their medicine and their physiotherapy on the same day, and they actually don't leave our institution until they're strong enough and fit enough to go straight back to school. Because I believe that it's really unfortunate. It's not their fault that they were given this disease, but they deserve a right to play, they deserve a right to go to school. school, to have an education, to have a life, and to be able to join in with their family. And generally now, that's what we're able to achieve in young people with myositis. I became a part of the TMA 6 years ago, and this is unfortunately my last year. I have had the most amazing journey with you through this conference and learned an enormous amount from all of the delegates, which has enabled me to go back and think about my work with the young people, because you have a voice and you have an ability to understand how this disease affects you, and the young people often haven't had that cognitive insight. So I've been able to learn and listen to you and be able to take that back into my own work. So I can only thank you enormously for the knowledge that you've given me. Thank you.
Thank you. Dr. Georgiou.
Good morning. My name is Wael Georgiou. I work at The Ohio State University in Columbus, Ohio, not very far from here. My journey with myositis started at the University of Virginia about 15 years ago. I saw this most amazing woman that was developing a very, very aggressive myositis after exposure to a statin drug. So that intrigued my interest. I took care of this patient for many years until unfortunately she passed away in spite of very, very aggressive care. That really left a major sort of change of direction in my career, and I did not study myositis before that time at all, and I started studying myositis since then. I'm an immunologist and a rheumatologist, and my focus is on membrane injury in myositis. So we have several years ago identified that if a cell membrane is damaged in animals, it predisposes them to myositis. And we've been studying that, and most recently we've started looking at people with myositis. And it appears that some of the autoantibodies that you've heard a lot about, some new autoantibodies actually affect the ability of a cell to seal itself. So essentially, the cell spills its content in patients with myositis, which aggravates the immune response and makes the patient have more and more severe form of myositis. So we think this is not just an observation that just is there for information's sake. It's actually a way maybe in the future we can target membrane repair in a therapeutic way that can enhance the treatment of myositis. But this is something that will have to happen many years down the road when we can prove that this can actually first work in animals and then we start in humans. Thank you.
Thank you. Dr. Ernst?
Good morning. My name is Florian Ernst. I work at Mayo Clinic in Rochester, Minnesota. I've been working there for 10 years and did my training in residency and fellowship as well. My journey began about 15 years ago when I started doing research in inclusion body myositis. And from there, I've gone into other research involving treatment trials with dermatomyositis and polymyositis. I'm part of the myositis clinic in Rochester. I see a couple hundred patients a year with dermatomyositis and polymyositis and inclusion body myositis. So I am primarily clinically based, and this is my second year at TMA. I'm happy to be here. I'm excited to be here. Thank you.
Thanks. Dr. Nagaraju? Oh, I'm sorry, Dr. McHugh.
Thank you. My name is Neil McHugh. I'm a consultant rheumatologist. I went to medical school, did my rheumatology training in New Zealand. So I'm a keen rugby follower. I follow a team called the All Blacks. My introduction to, I'm always been interested in autoantibodies, right from my early years in rheumatology, initially in the field of scleroderma. I spent some time in Australia and Melbourne learning techniques that I then followed up in my career in Bath in UK. I had a postdoc time at Yale University where I picked up other techniques that in fact came very handy in sort of measuring some of these autoantibodies in myositis, and hence in the last, I suppose, 10 years or so, we've had an interest in measuring autoantibodies, discovering a few new autoantibodies in myositis, and that's really been a major research advance in the field in terms of some of the discovery of new autoantibodies. But one of the challenges is getting these assays out so that they are more widely available in routine practice, and that's been somewhat frustrating in terms. And so one of the challenges is making available reliable sort of assays for measuring some of these autoantibodies, and that's a real challenge and one we're trying to address right now. Thank you.
Thanks. Dr. Shea?
Hi, yes, my name is Perry Shea. I'm at UCLA, Los Angeles. I remember very distinctly seeing my first myositis patient when I was a medical student, and it did leave a strong impression on me. I saw my first inclusion body myositis patient when I was early in my residency, and it was really inspiring to actually, I was inspired to go into the field of muscle, and particularly muscle myositis, because it seemed like an area where a lot of people talked about it, and there wasn't actually a lot of knowledge, or among a lot of the clinicians, it was something that people talked about, but wasn't very, there wasn't a lot of expertise in. So I did a fellowship primarily in muscle disease, and saw quite a lot of myositis patients, saw the spectrum, patients who were relatively easy to treat, patients that were difficult to treat, patients who were refractory to treatment, of course, and then the myositis patients that were not treatable, at that time. So I've become interested in education, in treating and training future doctors that were interested in muscle disease and myositis. I'm primarily a clinician. I see a lot of patients with muscle disease, and I participate in clinical trials, and I'm interested in outcome measures, measures that are important for function, measures that are important for our patients.
Thanks. Now let's talk to Dr. Nagaraju. Thanks.
Now it's my turn. So I'm kind of a little bit of an oddball here. I'm a veterinarian by basic training, and I did a PhD in immunology. And some 23 years back, I met Dr. Paul Plotz at NIH, a rheumatologist who significantly contributed to our understanding of myositis. He introduced me to myositis. Since then, I have been working on the myositis. Mostly initial stages, I spent quite a lot of time generating mouse models. As you all know, myositis, if you read the books, is the immune system that targets your muscle cells. So I want to understand to what extent immune system is responsible. Is it 100% or 50%? If it is 50%, what the other 50% responsible for muscle damage and weakness? So it's a little bit easier to do these experiments in the mouse models so that we can identify drug targets. So some 10 years back, as most of you in one form or the other have seen the drastic side effects of prednisone, and my lab has identified a prednisone that is safer with less side effects. So this drug has been now tested in children with DMD. So now it is waiting for an FDA approval. So we all can be probably, if FDA approves, so you have access to something that is better than prednisone. So I've been busy doing both lab as well as doing the entrepreneurial aspects of developing drug development. So that's my journey so far.
Thank you. Dr. Benveniste?
Good morning. My name is Olivier Benveniste. I'm coming from Paris at the Sorbonne University. I'm heading the Myositis Center in Paris, the only one in France. That makes that we have one center for 66 million of inhabitants in France. So a lot of patients are referred to us. And also we are doing some research, basic research, on, for instance, the effect of interferon type 1 on muscle and vessels, because this is the pathway involved during dermatomyositis, and we try to block this pathway JAK inhibitors, so we are working on that. And also we are working on IBM, and we just accomplished a trial using rapamycin versus placebo with interesting first results. And now we are trying to mount one confirmatory trial, with much more patients and many centers.
Thank you. Dr. Lloyd?
Hi, my name's Tom Lloyd. I'm a neurologist at Johns Hopkins Hospital. Really my interest and journey in myositis began as when I was a neurology resident at Johns Hopkins. I had actually research training in motor neuron diseases, especially ALS. And as a fellow, it was actually right after actually Andy Mammon and Lisa Christopher Stein had founded our Johns Hopkins Myositis Center. And so as a neuromuscular fellow, it was my job to not only see patients, but actually read every single muscle biopsy. And so it was through actually seeing patients and looking at muscle pathology that really made me realize how little we really understand about what causes IBM. And so a significant part of my lab now is focused on inclusion body myositis. In particular, we're actually working on developing a xenograft model in mice where we take actually human muscle biopsy samples and implant them into immunodeficient mice. And so we're really hoping to use this animal model to learn more about what causes IBM, and then also as a model for actually testing therapeutics.
Thanks. Dr. Alexanderson?
Good morning, everyone. My name is Helena Alexandersson, and I'm a physical therapist and a researcher at the Karolinska University Hospital in Stockholm. I got interested in myositis when I came to the Karolinska and about 20 years ago. I did see a few patients, and one of them asked me, am I allowed to brush my teeth myself? Because, well, my doctor said I should rest, so I don't know how much I can do or how much I should do. So that got me really intrigued, and I saw a few more patients, and by chance, those were pretty similar. They had muscle weakness, and some had the skin involvement. But then as I saw more and more patients, I really understood how diverse these diseases are, and that got me really interested. And then Professor Lundberg, sitting to the left of me, she also, she had questioned this notion that exercise was harmful. So she invited me to go into research, and we developed an exercise program, and we could prove that it was safe, and it could improve muscle function. And encouraged by the results, we got into more intensive research. And so I've always been interested in what happens in the muscle when you exercise. How can exercise improve the health of muscle? So I have continued on that journey. So I do exercise research. I have also focused on how could we measure muscle function in a good way, and how can we improve the patient-reported outcomes so that they really more reflect the patient perspective of the disease. And I see patients in the clinic polymyositis and dermatomyositis and inclusion myositis. So I see quite many patients each year, but it's really inspiring and wonderful to be here. It's my 5th year on the board, and it's great to see all of you and learn from you.
Thank you. Dr. Mozaffar.
Good morning. I'm Tehseen Mozaffar. I'm a neurologist. I'm at University of California, Irvine. And this is my 2nd time on the medical advisory board. I was on the board for 6 years prior, including 2 years as the chair of the medical advisory board. My interest in myositis started back in 1995 when I started my fellowship in neuromuscular medicine under Alan Pestronk at Washington University. And we got interested in looking at various forms of immune myopathy, but most importantly, looking at Jovan myositis. And we were the first ones to define the muscle pathology in Jovan, which we reported as different from other forms of myopathy. And subsequently, this work has led to other groups confirming the findings that we reported back in 2000. And so it has led to the concept that antisynthetase syndrome may be different from polymyositis and dermatomyositis in terms of clinical features as well as myopathological features. And for the last 10 years, I've been very interested in IBM and other myositis. Dr. Goyal and I were the first ones to report differences in severity of the disease depending on whether you have the antibody for IBM or not. We reported that about 2 years ago. And again, that work has been confirmed in a European group study as well, suggesting that there may be differences in not only muscle, in terms of clinical picture, but also some of the muscle pathology pictures and disease progression, whether you have antibodies or not. I'm currently focusing on better defining some of the immune cell abnormalities in IBM with the specific target of developing clinical trials or drug treatment trials for a specific type of immune abnormality that happens in IBM. And we are also trying to validate one of the scales that has been developed for measuring dysfunction of the bulbar muscles in IBM, so we can use it for clinical trials in the future.
Thank you. So Dr. Lundberg is next, and I just wanted to say that she's not a member currently of the TMA Medical Advisory Board, but she's here for a special award that we will find out more about at lunchtime. So thank you for being here.
Thank you, and good morning. I'm Ingrid Lundberg. I'm a rheumatologist from the Karolinska University Hospital in Stockholm, Sweden. And my research in myositis has really been inspired by meeting patients. And we go back to the 1980s when I started in training in rheumatology. And at that time, according to textbooks, the treatment was recommended to be based on steroids, immunosuppressive agents, and rest, as you heard. And the effect was slow. But one day in my office came this man on a follow-up visit. He was very happy, and he proudly showed me, laying down on the floor in my office, how many push-ups he had learned to do since he last met me. And he was so pleased because he started to train with his daughters, who were elite swimmers. So, and then it happened a couple of weeks later, there was a young man, an ice hockey player, who also was so pleased because when he had gone back to play with his team and train with them, he did so much better. So then I was sort of confused because according to the textbooks, you should rest. And I started to go back in the literature to find the reference for the recommendation on rest. And that recommendation I found was based on biopsies, studies of muscle biopsies on marathon runners. So after there were studies where marathon runners had been subject to muscle biopsy, and they had a lot of inflammation. So then the conclusion had been that if you already have an inflammation in your muscle, exercise may worsen this inflammation. And then that had led to the recommendation of rest. So and then, inspired by that, I thought we have to find out. And then I was very lucky to meet this young physiotherapist, Helene Alexandersson, and recruited her to a project, and we applied for funding, and that is how the research started, and we managed to, in a very careful way, test if exercise was safe, and also it turned out to be beneficial. So I think, and I've also been on this board for some years, and it's really inspiring, and I think the meeting with you who know about the disease has been so important for me, and also then for the meetings in the clinic. So we learn from you, and I really thank you for all support also that have been given from you. Thank you.
Thanks. Dr. Ryder is also not currently a member of our medical advisory board, but she's also here for the awards.
Okay, thank you. My start in myositis began in my fellowship years. I came to the National Institutes of Health and worked in the lab next door to Paul Plotz. And I came to patient rounds and learned about adult myositis on rounds, and I thought that adult myositis sounded a lot like juvenile myositis when he talked about different autoantibody subgroups. But he told me those autoantibodies did not exist in children and that the disease was very different. So we decided to start a project together, and I had been training in Seattle prior to the NIH, where I saw a number of myositis patients. And we took their sera and tested them and found those same autoantibodies in the children as in the adults. So Paul then introduced me to Fred Miller and asked that I take a position with Fred next, which I did. And Fred assigned me 2 projects. One was to understand the differences between children and adults with myositis and about these autoantibody subgroups. And the other was to develop tools that we could use in the clinic to assess patients' treatment responses. And that work started, in fact, Ann Reed was one of my first collaborators in that project, a multi-center project to develop those tools, and that work evolved into what's known today as the IMAX group, the International Myositis Assessment and Clinical Studies group, where we have gone on to develop response criteria for treatment trials. So I'm still working on those 2 projects today. Those have been the projects that have defined my career. And I've gone on to do some other studies related to treatment trials as well. And I've also been very involved with the patient support group through the years from the beginning with this group. It was a pleasure to also serve on the medical advisory board for a number of years. And more recently, I've been working behind the scenes with this group, particularly on the MyoVision repository study, and I hope that we'll have a lot of work coming forward on that study. Some has begun to come out. Thank you very much for all your support.
Thanks. And we have Dr. Yetterberg.
Good morning. My name's Steve Yetterberg. I'm a rheumatologist at Mayo Clinic in Rochester, Minnesota. I'm currently the medical advisory board appointee to the board of directors, and I'm proud to serve in that capacity. I'd point out first that people often ask me where my name comes from. It's Swedish. All of my relatives are Swedish, so I think that's why I was seated in the Swedish section, not of the panel. But I assume it was planned. People have talked about patients inspiring them. I actually came to myositis research because as a fellow, I started in a research fellowship and ended up in a virology laboratory at the University of Michigan. And one of the things that my mentor was studying was a particular virus that seemed to cause muscle disease. And so we investigated that for some time. So I sort of got into the field because of the work I was doing in the laboratory, and that spread to seeing patients. Fortunately, I was recruited to the Mayo Clinic about 20 years ago because before that I wasn't seeing many patients, and since being at Mayo I've had a chance to see many patients. It's a wonderful place to work. I think I'm blessed to work there because I have access to many colleagues who have expertise in many fields. My office is right next to Dr. Ernst's. I am primarily a clinician at this point and mostly see patients and involved in some clinical research. It's a pleasure to run into patients I've seen here. It's a pleasure to have met other patients, and I agree with everybody else that you all are inspiring and keep us going here. Thank you.
Thanks. Dr. Goyle?
Good morning. My name's Namita Goyle. I'm at the UC Irvine. And my career actually started at Mass General in Boston, where I had done my fellowship, and I stayed on faculty there for a few years. That's probably where the greatest impact for myositis began for me. I met a patient who was deteriorating. I needed a muscle biopsy to make an accurate diagnosis, and the surgeon, had him on the patient on her list to do the biopsy, but it wasn't a priority for him. And so over the weeks, I was watching the patient deteriorate, and I had to really urge and plead the surgeon to get a biopsy on her. Unfortunately, the patient had a poor outcome, but I learned from that experience early on, and I actually made a deal with the surgeon, and I said, I will do all the surgical biopsies for myositis patients at Mass General if you just train me. And I just learned early on that early diagnosis and management for our myositis patients, we can actually make an impact for them. And then my other focus has really been clinical trials. When I was in Boston, I worked with Tony Amato and Steve Greenberg on the original bimagrumab study in IBM. And then when I moved to UC Irvine, when the study had moved forward with the large worldwide study in bimagrumab, I also was an investigator on that. And through all my clinical trial experiences, I now lead the clinical trial unit at UC Irvine. And I've learned that it's really important to design clinical trials well, to take the time to make sure that we are assessing the right measures. So we won't be able to find a drug that's effective unless we're using the right tools, which is where outcome measures have become very important. And so I spend my other time on finding better therapeutics for our myositis patients.
Thanks. Dr. Weil?
Good morning. I'm Chris Weil. I'm at Washington University in St. Louis. I'm a neurologist by training, and I guess the way I see my career is I straddle two different lives. I see patients in clinic, but I also run a basic science lab, and I consider myself what's called a translational physician. So I am interested in understanding things from a molecular and cellular aspects and how we can translate those into therapeutic targets so that we can move those then into the clinic. And by having my feet in both of those areas, I'm able to see the needs of the patient and understand the needs of the patient, understand what's going on from the clinical aspect, and then also be able to look at that from a more reductionist type standpoint in the lab. I'm particularly interested in genetics of myositis. I'm interested in age-associated weakness, such as happens in inclusion body myositis, and then interested in muscle degeneration and how we can see if we can combat that for some of these age-associated myositises. And finally, I want to thank you guys, just as everyone else has, for inspiring me and keeping me thinking and asking questions so that we can work together to cure these types of diseases. So thank you.
Thanks. Dr. De Visser?
Good morning. My name is Marianne De Visser from Amsterdam. I work at the Academic Medical Center, and I'm a neurologist by training. My journey started in the laboratory of Dr. Andrew Engel in the Mayo Clinic in Rochester, Minnesota. He was and still is an expert on myositis, and he assigned a project to me to investigate the tiny blood vessels in the skeletal muscles in dermatomyositis. So I spent quite some months there, and when I returned to the Netherlands, I noticed that neurologists were not involved at all in the care of patients with myositis, other than reading a muscle biopsy. But that gradually changed over the years. I became a member of the multidisciplinary team because we know that myositis is more than only muscles which are affected. And at that time, also my research started. We did studies on the natural course of myositis. We also looked at differences and the commonalities between the various subtypes of myositis, and I became involved in and also initiated treatment trials. In the Netherlands, I'm medical advisor of the working group on myositis, and I consider it a great honor to be a member of the medical advisory board of the Myositis Association. Thank you.
Thank you. I'm going to change it up just a bit and invite Dr. Mammon to speak.
All right, thanks. So my name is Andy Mammon. I'm a neurologist by training. I did my neurology training at Johns Hopkins and then decided to do a neuromuscular fellowship where we learn about nerve and muscle diseases, and I didn't have a very strong direction at that point in terms of what I was going to do in the future. And as part of what we did, as Tom explained, was to look at a lot of muscle biopsies. And I have to say my original interest in myositis came through looking at muscle biopsies and just being fascinated by them and trying to learn more about it, the disease, learning that there was not that much known about what were the causes of myositis, how to treat myositis, and so forth. So I got quite interested in that and decided that would be a good thing to pursue for my career. And fortunately at Hopkins I ran into some like-minded people. So Lisa Christopher Stein was a rheumatologist, also just kind of starting out her career trying to study myositis, and Sonia Danoff, a pulmonary specialist who was interested in the pulmonary complications, lung problems that many myositis patients have. And so we decided to get together and start a multidisciplinary clinic, sort of in the spirit of others who had done the same thing in the past at Hopkins. And it started out with the three of us back in 2007, trying to provide really a multidisciplinary approach, not just to taking care of patients, but also bringing our different perspectives to the research. And that center has grown. We have Tom Lloyd in the center who you've already met. And then it's grown to be about 10 full-time faculty who are really focused on seeing myositis patients. And I would say that, although my original interest sparked from, looking at muscle biopsies, a lot of my research since then has really been driven by my interactions with patients. And just sort of one example of that, we were interested in patients who have a particular type of myositis called necrotizing myositis, where there's a lot of muscle fiber damage in the muscle but not a lot of inflammation. And some of the patients who we saw who had this type of muscle biopsy and myositis were telling us that they thought statins might have originally caused their muscle problems. And at first we were kind of dismissive of this idea. And in particular, I didn't think that was really probably the case, but more patients kept telling us that that seemed to be a common theme, and so we decided to do a study, and lo and behold, our patients actually knew, what they were talking about, and since then I've learned, to really listen to patients because they often know more about their disease than I do, and we really learn from you, and that's really just continued to be true, even in going to these the patient conferences. I think the idea is that we're supposed to impart information to you at these conferences, at these small group sessions, but often I get asked questions that really make me go back to the drawing board and think about myositis in a new way, or try to learn something that was sparked by a question somebody asked. So since 2014, I've been at the National Institutes of Health, where my research group is, and we continue to try to understand what causes myositis, what the sort of basic mechanisms are, what the different types of myositis are. Maybe they need to be treated differently, and it's just really been a rewarding journey that I've been taking with the rest of you guys. So thank you very much.
And finally, Dr. Rohit Agarwal.
So they keep best for the end. Okay. Well, my name is Rohit Agarwal. I'm a rheumatologist by training. I'm at University of Pittsburgh, Myositis Center. I got interested in myositis purely because of my mentor, and that really tells the reason we need to encourage younger people who, some of us now can mentor some of the younger people into myositis field and do more research into it. So my research started in myositis purely because of my mentor said this is a great opportunity, it's a great disease, and he was right. And since then, I have really enjoyed taking care of amazing patients, and it's this enigmatic disease. What I've done in the last 10 years is focus mostly on clinical trials and outcome measures. And what I'm planning to do for next maybe 5 years is trying to develop this direct-to-patient approach using smartphone, wearable technology, telemedicine principles, where I'm trying to go directly to the patients to collect the research data and do more meaningful research. As you know, because of the rare disease, access to patient is a major problem, but we are so many patients in the country, so I was thinking I'm trying to develop this approach which, if successful, really can be the game changer in research in myositis. So as a part of that effort, I got an NIH grant recently where I'm trying to develop a mobile app and a study website to enroll patients and follow them over time, send them Fitbits, do remote testing on them, do telemedicine approach on them. So there are a lot of stuff going on, and I talked to you a little bit about that study earlier. So as a part of the study, I would like to know if you could, by the show of your hands, tell me how many of you have iPhone? Quite a many. Yeah. How many of you have Android? Quite a few. How many of you don't have Android or an iPhone? That might be easier. Okay, so some. All right. So we need the people who don't have Android or iPhone. Would they have access to a website that they could use, share their data or story? Okay. All right. And then how many of you would have a data plan or internet access to share your data from your home? Many of you, all right? And how many of you would prefer a mobile phone or a smartphone to share data as compared to website? So smartphone maybe, and website now? So some would prefer, so we need to definitely have both approaches. All right, and how many of you will be willing to wear a Fitbit if sent to you for free for research data collection? All right, that sounds good. And last, but it'll really help me, is how many of you have access to your electronic medical records? Quite a few. Excellent. So that means there should be no reason that everybody sitting in this room, or most of you sitting in this room, should be joining the study when it gets launched. It will be promoted through the Myositis Association email and their website. So I really look forward to see you in my research study. Thank you very much.
Thank you, everyone. So this is the time. We have about 50 minutes where you all can ask your questions of our distinguished panel. In order to be able to get as many questions in as possible, we'd like you to keep your question to a question. And just to remind you that a question is a sentence with a question mark at the end. It takes very little time. So, okay, so we have some questions. We'll go back and forth. Go ahead.
I'm raising my hand for my husband to raise his arms.
This is for the dermatologist. I have IBM, and you can see it's pretty advanced. But my fingers seem to be very sticky all the time. I mean, if I hold a fork or anything, it's like glued to my hand. If I put my fingers together, you can feel stickiness. Anything I touch. Any ideas?
Maybe you guys probably know this, but dermatologists, we really like to look at things. So you know what I'm going to do as soon as this meeting is over is I'm going to run down and look at your hands. You know, sort of what you are describing, Probably not something maybe particular to your muscle disease, but we have patients who have essentially what we call hyperkeratosis, or just sort of abnormally scaly or flaky, thick skin on their fingers. And in some case, patients really find that to make their hands really sticky when they're trying to do tasks. And sometimes actually we can use special creams, so not things that you can get just from a regular moisturizing cream, but that actually helps smooth that skin down so things won't feel as sticky and therefore as tacky when you're trying to do tasks. But if you don't mind, I may run down and take a peek at your hands as soon as we're done. Okay, thanks.
Over here.
Thank you. To everybody on the panel, I'm wondering if you all have used a drug called prednicort. I met a polymyositis patient a few months ago, and she's being treated by a doctor from France, and we were comparing and contrasting notes, and she was far better than I am. So I was just wondering, and I did some research, and from what I read, it's not used by doctors in the United States, but in some other continents. And shout out to Dr. Ryder.
Dr. Benveniste, do you think you can answer that?
What is your question? I don't know. Where are you?
So, can you ask her to tell us what the name of the drug is again?
Prednicort, prednicort. Prednicort.
Yeah. They've all got their smartphones on and they're looking it up. So I think the answer is no. No one here has ever used that.
It sounds like steroid, yeah.
Yeah,
so prednicort is the same medication as prednisolone. In Europe, they like to use prednisolone. In the US, we use prednisone. They're the same identical drugs pretty much.
Great, thank you. Where are we over here? Okay.
Hi. I have two questions. One is for Dr. Agarwal. I did read your RIM study, and then I think there was a follow-up, and I'm just wondering if that the results in that study had changed the way you prescribe rituximab in your patients with myositis.
So, yeah, I mean, I think RIM study actual trials results were negative, but we learned a lot since RIM study, and there have been subsequent many publications by our group as well as other groups. So it has definitely changed my attitude towards rituximab. We use rituximab primarily still use it for refractory disease and interstitial lung disease, and especially used in a subset of patients who have this antibody called Jo1 or anti-synthetase antibody. At least in my experience in that antibody, it does stand out significantly better than some of the other antibody. It does improve other patients when they are refractory, but for anti-synthetase, that's generally my second or third drug of choice after they fail the basic treatment.
And just as a follow-up to that first question, what are your thoughts about it in terms of refractory skin disease in myositis?
So we have a publication from the rituximab study on the skin disease, and I would say the results were positive in terms of it seems that there was a positive response. You can't, because the way the RIM study was designed, I cannot say for sure it made a lot of difference or not, but clinically when we have used it, we have seen moderate benefit.
Okay. And then just to the, thank you very much, and then
I'm
just
sorry,
to the panel in general.
we really need to ask somebody else. I'm sorry.
Ask?
Jim?
My name is Karen Shaw. My husband has IBM, and I have a question for Dr. Mammon. In regards to the studies on the statins, my husband is still on a statin, and so is there any benefit now to stopping that, or
Yeah,
what are your thoughts on that?
Yeah, so we think that most patients with inclusion body myositis have no greater risk for side effects from statins than other patients, and patients who have a medical indication for being on statins should be on statins regardless of the inclusion body myositis. It's only a very small percentage of patients who have statin-triggered autoimmune muscle disease, we think, that need to avoid the statin medications, and those would be the patients who have the necrotizing myopathy with the anti-HMGCR autoantibodies. As far as we know, for the rest of myositis patients, statins don't pose a unique risk.
Thank you.
Hi, I have two questions. We were sent home from Johns Hopkins with no help for IBM for my wife, but yet everybody sees rheumatologists, neurologists, and am I missing something that I can't help my wife with? And the second question is, we've seen on TV about using ketamine to help people with IBM. And when I researched it, I find there's a lot of doctors, neurologists throughout the country that are using ketamine. And so we have an appointment in October with a doctor from Penn Hospital. Have you heard of it? And what do you think about that?
Dr. Lloyd, can you take that?
Sure, yeah. There are a number of places that are using IV ketamine for a variety of neurological and neuropsychological diseases. And I'm actually familiar with a few places that will advertise IV ketamine infusions for an incredibly long list of diseases. I'm not aware of any rationale for actually trying ketamine in inclusion body myositis, and certainly there's no data. Yeah,
I think the bigger concern with ketamine, and I think Dr. Lloyd is absolutely right, I mean, the data in support of ketamine is really not there. And there are significant risks, including risk of death, that is associated with IV ketamine. I mean, it's been known that if they are not monitored, IV ketamine can cause significant harm. So I'm not sure that it's necessarily the way that I would recommend in that. But to answer your first question, I mean, we do not have a cure for IBM. We may not have effective treatment for IBM. That doesn't mean that the patients should not be followed in a myositis center or with specialists, either rheumatologists or neurologists who specialize in IBM, because we can do a lot for these individuals, whether it's taking care of their disability, whether it's taking care of some of their symptoms. So I think it's still important to follow up on a regular basis, irrespective of whether we offer cure or treatment or not.
Thank you. Terry?
I'm not sure who to ask this of, but I was wondering if there were any data as to spontaneous remissions or abatement of symptoms that have been documented, and what might be the factors to that.
In which form of myositis?
Any form.
Okay. Anybody?
So I think not in IBM. Among patients with autoimmune diseases, some patients seem to have what we would call monophasic disease. One episode of it, and it quiets down and goes away. And that occurs occasionally, and basically you don't know until people get better with treatment and you start backing off the treatment, and if it doesn't come back, was that a remission? It's hard to know.
The other areas where I've seen spontaneous remission is when few patients who have cancer-associated myositis, as the cancer gets completely cured, I've seen few cases where they've gone into spontaneous remission, although I would say most patients still need treatment for myositis, but a couple of patients who have no treatment of myositis but gone into spontaneous remission after that.
Thanks. Lori?
Oh. Hi. My name's Dave Volk. I have IBM. And I guess this question, I'd like to direct it to Dr. Weil. Dr. Weil's a physician I go to at Washington University. And I have a couple of questions. One is regarding the diagnosis of IBM. I had a muscle biopsy and confirmed the diagnosis, but I also am very familiar with WashU and their Center for Clinical Imaging Research, which is one of the best imaging research centers in the country. And I know that MRI, CT, PET scans are getting more advanced, and I'm wondering if there is thought about using that as a better diagnostic tool that's less invasive and gives you information with regard to IBM, and also establishing a baseline for all myositis patients to monitor the progression of this degenerative disease that we have. And on top of that, I know also WashU has one of the best data centers for mapping the human genome, and I don't know if that's being utilized to identify markers or in the human genome that might identify aspects of this disease. Thank you.
Great. Thanks, David. I haven't seen you yet, but hello. And hello to your wife as well. So the two questions were, the one was about using imaging modalities to enhance diagnosis as well as to enhance perhaps understanding disease progression in diseases like inclusion body myositis, but we could even generalize that to other myositises as well. And imaging technology is getting better and better, and we're starting to understand how to use these different imaging modalities. And the benefit of an imaging modality, where I'm talking about an MRI or a PET scan, is that they're non-invasive to the patient. They don't require anyone to do a muscle biopsy on the patient. And so we do MRIs and different imaging modalities to help us, and this is going to sound the exact opposite of what you're saying, but help us to identify where we should do a biopsy so that we can actually identify an area of affected muscles, so that whenever we do a biopsy, we're actually doing a region of the muscle that we think is affected, because the last thing we want to do is do a muscle biopsy that isn't helpful. So that's one reason we would use an MRI. But your question is more about, can we use it to diagnose? And I think we're moving in that direction, where there's certain patterns of weakness that we can detect on an MRI that in the right clinical context can help us diagnose a patient with IBM. And so I think that's absolutely true. The issue is, at least in the United States, it's easier for me to get insurance to cover a muscle biopsy than it is for them to do an MRI. And so that's part of the reason that we tend to move in that direction. That's different in other countries where MRI is used more readily. The second aspect to your question is about whether MRIs can be used to understand disease progression. And I think that's a great question that is being studied at many different centers. And the idea would be, could you use an MRI to let someone know if they are getting better on a therapeutic intervention? Could you let someone know if they're getting worse, and would there be something you would do different? And so I think those are coming. Whenever we look at an MRI, there's many different things we can look at. We can look at just not the size of the muscle, but we can look at the kind of characteristics inside of the muscle, so the inflammation that's inside of the muscle, the amount of fibrosis that's in the muscle. And so what we need to do is to really start carefully looking at those things longitudinally. But we would need to characterize those things longitudinally, as well as doing an assessment of how the patient is doing, because getting information that tells me that your MRI is getting worse means nothing unless I can tell you that you as a patient are correlated with that. So those are things called natural history trials, where we try to use things like the clinical outcome along with a biomarker, and the biomarker would be something like an MRI. And then to your second question about genetics, we're very interested in understanding the genetics of all of these diseases, and it just comes to getting as many patients enrolled, getting good clinical information on those patients, doing the studies, and then correlating them with large bioinformatics type studies, and we're doing those. Those are really exciting things that I think will yield information.
Thanks. Let me just remind everybody that we need to keep our comments to a single question, please. Jim?
This is a question for Dr. Mammon. I participated in a study at NIH in December of 2014. I'm not sure if you were there at the time. Into the general causes of dermatomyositis. I haven't had any follow-up. I'm just curious if you know if there are any results from that study. It may be ongoing still.
So I actually wasn't part of the whatever study I think you were involved in, so I can't really speak to that. But if you want to meet afterwards, I can learn a little bit more about why you were there and who you saw. Did you see him?
No, I didn't, but maybe I can speak to it.
Oh, sure, yeah.
Do you know which study you were in at the NIH at the time?
Well, I think it was just a general study. Dr. Mammon mentioned the PI. It was in an earlier comment. If you can, I don't remember his name.
Was it Dr. Plotz?
No.
Dr. Miller?
No.
No. Dr. Katz?
No.
No.
Well, Dr. Mammon mentioned his name, I think in another presentation or maybe this morning. It was.
Did you see Dr. Schiffenbauer?
Yes, I did.
Okay, so you were in Dr. Miller's study. We have a number of ongoing studies looking at environmental and genetic factors. We had a Meyer-Risk study enrolling anti-synthetase patients and other patients early on in disease. We have a military myositis study. We have a twin sibling study of twins and siblings discordant for disease. So all these studies are still enrolling patients, and there are some publications forthcoming, but they aren't completed yet. And we also don't release results directly back to patients, but through publications, and often TMA covers those results in their newsletters.
Yes, mine was just for DM only.
Okay. It's probably one of those studies.
Lori?
Hi, my name is Sue. I have IBM. I was just a quick question. Does anybody on the panel know how many cases are diagnosed each year on PMDM and IBM?
I don't think we have that data. Terry.
I have dermatomyositis, and in one of the presentations, one of the doctors mentioned that there is a corrosive or arthritis type thing that in your hands and that can come with dermatomyositis.
Was it an overlapping complication?
I don't think so. They said that.
Which presentation was it?
See, I can't quite remember. There's so many. I think it was with Dr. Goya or
Goyle?
Yeah. But it could have been
Perhaps
Mammon.
we're talking about the anti-synthetase syndrome patients where there's a constellation of symptoms, including interstitial lung disease. There can be some arthritic changes to the hands.
Yeah.
Is that
Because I haven't been diagnosed with anti-synthetase, but I have that problem with my hands, so.
Lori?
Is there any interest or research being done into the use of good old marijuana, CBD, whatever you want to call it? And the reason I ask is because someone recently told me that they were using it because their doctor said it would be good for their inflammation that they had. So I'm just curious, is there any thought of using it for anything to do with myositis?
Dr. Vlugel, do you think you can speak to that at all? Maybe not. Nobody?
So, I mean, as far as
I'm going to just mention stuff from the skin perspective, and then I'll pass the microphone down if other people want to talk about it. So there are many people using this in various ways for many conditions, and some in the skin, some of the things can help with inflammation and itch. So in various conditions, we have patients, we have other approaches to using various forms for both skin and itch. And then also there are many of my colleagues using it for other diseases, and I'm going to pass it down so people can comment on those.
I mean, at least what we know from myositis inflammation, there's no evidence of that, at least in pure myositis. Now, if you're talking about treating pain and myositis-associated pain, muscle pain, or overlap with fibromyalgia, I know there are people who are using it, but still the evidence is pretty slim on that.
There is a clinical trial completed, a phase 2 trial of a drug that's derived from the THC, and it's not exactly the same product, but a derivative product, and it looked like, we haven't seen all the results yet, but it looked like it had promise for patients with dermatomyositis, particularly for skin disease and the itch. And Corbus Pharmaceuticals is planning a larger, more definitive trial for dermatomyositis. So we hope to have more results in the future.
And Corbus is here as an exhibitor, so you might want to stop by that table. Jim?
I had a question more on immunology. I see and hear a lot of research on looking at the muscles, but is anybody looking more at the T cells and B cells that may be triggering this off?
Raju?
No, I think a lot of people carefully looked at the role of T cells and B cells. We know that the B cell role is clear because most of these diseases have autoantibodies triggered and very specific to myositis. So, but we don't know, one of the unique features in this disease is nothing is muscle specific. Almost all the antigens these antibodies react to are ubiquitous. That means every cell has. And also very early on, people showed T cell role. One of the roles people predicted is that you have cytotoxic T cells going and damaging the muscle fiber. The issue is muscle is 30,000 times bigger than a cytotoxic T cell. To what extent the damage occurs because of T cell mediated killing? We still don't know. So people looked at carefully and continue to look at these two cell types.
Can I also?
So maybe could I also comment here?
Sure.
As we are looking for specific T cells, mainly in the blood, but also we have looked in the bronchoalveolar lavage fluid in the lungs, in particular in patients with the anti-Jo1 antibodies in lung involvement. So we have found some antigen-specific T cells, so we're working on that. So there will be hopefully publications coming out on that soon.
Great. Good. Yeah, and there are multiple studies looking at all forms of myositis, and particularly seeing different types of T cells, B cells, and also cells called dendritic cells that are in multiple tissues, the skin, the lungs, the muscle. And all of those cells, and it varies from subtypes slightly, they produce different proteins that we also think are now affecting potentially the tissue breakdown or the other additional inflammation in the tissue. So there's quite a few studies looking at that.
First, I want to thank all of you for all your hard work and dedication to all the help in myositis. My question before is, actually my father is more advanced than, and I hear a lot of here is kind of the beginning of studies and trying to help people early in stages, but is there anything for people who are more advanced, patients who are more advanced or who seem to be deteriorating quicker than others to help slow down that process? And anything that we can do as caregivers, any ideas that we can do to help make the anything easier, and I am also an All Blacks fan, so.
So did you say
IBM.
dermatomyositis or IBM?
IBM.
IBM.
So, although actual studies are lacking and are currently ongoing, our sort of anecdotal findings are actually patients who are more physically active, able to actually exercise frequently, as frequently as they can. It seems like those are the ones who have a slower rate of actual progression. Now, more studies certainly need to be done.
Anybody else? Okay, thanks. Terry. Terry.
My name is James L. Davis. I have IBM. My question is, I was diagnosed with carcinoid tumors before I was diagnosed with IBM. Now, in all your studies and research, they do not want people that has cancer. Why?
So I can try to answer that. So the reason in most studies the cancer is excluded because when you're trying to do a study, you only want to study a drug versus not a drug, which is like a placebo. So if there are other factors that could affect the disease, then you would not know would the drug worked in a particular patient or not. And cancer appears to be that one of that factor that can influence the disease significantly. So that is why cancer is, one of the reasons is why the cancer is excluded, is that to prevent any what we call confounding for the result of the study. The second reason it's excluded is because of the safety. So if you're giving a drug to there's a new drug, new molecule, and you're giving it to a patient with this disease, but we also know if they have cancer, underlying cancer recently, we don't know the interaction of that drug to the cancer, what it would do to cancer, could it reactivate the cancer? So that's another reason the safety of the patient is also kept in mind to exclude the patients with the cancer for that research study. So those are the two main reasons they are given that why cancer should be excluded. But once the drug is approved, then perhaps those patients can be given that drug to see if it works or not.
Jim?
My question is in regard to hereditary IBM. Are there any current studies being done on hereditary IBM? And as a corollary to that, will the studies and trials on sporadic IBM potentially have a benefit to understanding the hereditary form of it?
So that's a great question. So hereditary IBM is probably broken down into actually many different forms of hereditary IBM. So that's one comment I would have, and it's quite different from the sporadic forms of IBM. However, the molecular pathways and the molecular mechanisms very well may be similar, and so we firmly believe that studying the hereditary forms will inform us about the sporadic forms, and the sporadic forms will inform us about the hereditary forms. And in fact, for Dr. Domaczki's clinical trial, the actual study is being done in sporadic IBM, but all of the preclinical work, which means the work that was done in an animal model, was actually done on a rare form of hereditary IBM. So I think you're exactly right that research in both can inform both, and therapeutic trials, although initially trying to identify a specific subset, so the trial may only be for sporadic IBM, that does not mean that once that drug is approved, it couldn't potentially be used for something like hereditary forms.
My name is Bill Matthews. My wife has IBM. I have myasthenia gravis. Has there been any consideration among the board of looking to sponsor or encourage TMA to put together a repository of information provided by myositis patients online that can be used by researchers to determine information such as how many people have this particular type of characteristic versus another type of characteristic. This is what the MG family has, and it's very useful. Thank you.
So TMA had funding from the Centers for Disease Control to do a national myositis patient registry. Patients enrolled in this registry back in 2010 to 2012, 2,000 patients enrolled. Most patients didn't complete it online in those days. We offered that option, but only about 400 patients took up on doing it online. It was a very long questionnaire, 35 pages long. In fact, our group, Fred Miller and I, and our epidemiologist, Christine Parks, worked hard with Bob Goldberg to develop the questionnaire. And it was focused on general features of myositis, treatments received, a lot of environmental factors like infections, medications, occupation and hobby exposures, smoking, UV light exposure, and then a quality of life questionnaire. So the data has been analyzed, and some work is starting to come out. The work on quality of life has appeared in publication, and we've shown that myositis patients have significantly reduced quality of life compared to even patients with rheumatoid arthritis, and certainly compared to the general population overall in the US. And we will have other publications out on this, but it's not perhaps as detailed as you might hope, and certainly follow-up studies will be needed. And one of the things really missing from this registry was a control group at the same time to have enrolled. So it's a descriptive study that will beg new questions. And ultimately, I think it will be open for other researchers to use. We're going to, this next year, probably transfer the data to the National Institutes of Health so that researchers in the future can apply to use that data set.
Dr. Lundberg, do you think you can talk about the European registries?
Yes, we have an international registry for patients with myositis, but it's not at the moment open for patients to register their data, but for the physicians. But we are working on tools for patients to enter their data once they have been diagnosed and included by the physicians. So we don't have any registered like that patients enter themselves data that I am aware of.
Thanks.
Thank you. I'm humbled by all the experience and expertise we have, and I thank you. A couple of quick questions. With technology available to us today, is anybody gathering all the information from all the resources so that nobody is reinventing the wheel? And also gaining experience from the people in the audience who are more than willing to share what we've been through. And a Judge Judy question for Dr. Agarwal: will you allow Canadians?
So at this moment, it's only for US and only for dermatomyositis and polymyositis, but that's only because of the lack of funding. It's not because of the lack of the technology or lack of our intent or means. It's because of the lack of funding we had to limit it, but I'm trying to actively seek funding to expand it to a couple of things. One is inclusion body myositis, second is juvenile dermatomyositis. Third goal would be to include a larger North American population, and the next goal would be maybe international. So that they are all things are being planned, but just the way it's done.
Okay, I could add something about your question about reinventing the wheel and all the physicians trying to learn from each other so that we're not repeating and doing exactly the same thing. So in the past 15 years, there's been an international myositis group formed called the International Myositis Assessment and Clinical Studies Group. It started out with work on trying to develop response criteria so we would have measures to show that patients are responding to drug treatments and could be testing new drugs in clinical trials. And the group has expanded quite a bit. In fact, now in recent years, we're more than 250 myositis physicians and researchers and other specialists from around the world in this group. And there are projects being done by members of the group that are initiated by different scientists in the group. For example, Dr. Agarwal is leading a project on cancer and myositis. Dr. Asherman, who used to be very involved here in the medical panel, is leading a study on interstitial lung disease. Dr. Lundberg had a study on classification criteria that was also run in part through this group. So the researchers are talking quite a bit and trying to work together on new things.
I'm going to give just a quick dissenter's opinion on this. So from the context, so I hear what you're saying about not reinventing the wheel, but from the context of basic science, taking a fresh look at things, reinventing things is really part of what we do. And so while sometimes it feels like it's circular, it's really looking at it with new eyes, looking at it with new technology, and so not as much reinventing the wheel, but just getting a fresh look at it.
So my question is, how close are we to inventing a synthetic immunoglobulin, synthetic IgG?
I'm sorry, can you repeat the question? I'm not sure.
How close are we
IVIG.
to inventing a synthetic IG?
So there are at least 3 companies that have created what's called a recombinant immunoglobulin molecule, and these are not full or a complete immunoglobulin, but they are mainly the FC fragments, which is the main fragment that sort of creates the trouble in some sense. And the idea is that if these FC fragments would bind to muscle membrane and not allow the disease-causing antibodies to then attach to the muscle membrane. They're primarily being looked at in myasthenia gravis right now. So there are 3 different companies that are doing trials in it. So far, and I may not be aware of it, but I haven't seen anything in myositis, but I would suspect that there may be a trial of it in dermato first, which is one area where IVIG has a clear and distinct role. Necrotizing myopathy would be another area where these can be potentially used. But right now, the initial studies done by these companies are in myasthenia gravis.
Thank you. My question is for Dr. Musafra. I'm one of the 1% of statin users who came down with the condition, and it was proven that it was statins because when I went off of it, the main symptom that I had at that time, which was falling like every other day, disappeared, and I no longer had that. But now I'm told, because it was brought on by an external element, the statins, that it's VM. I'm also told that I'm PM, and how much of this stuff overlaps? And if it does overlap, how can I be determined that I'm on the correct treatment for what I have?
And who was your question directed towards? Dr. Mammon?
Dr. Musafra.
Yeah, I was actually going to defer it to Dr. Mammon since he's the expert on statin myopathies, but sometimes the clinical picture can be confusing between PM and DM, especially if the skin rash is not prominent. The other consideration, and especially from a heretic like me who doesn't believe a lot in PM, anyone above the age of 50 with diagnosis of PM, I would seriously consider inclusion body myositis as a possibility as well. But then the statin-related question, I would defer to Dr. Mammon since he's here.
So I would agree with everything that Tahseen said. The only thing I guess I would add to that is that we have a pretty good way of figuring out who has statin-triggered autoimmune muscle disease, which is testing for the antibody. And it may be that some patients with dermatomyositis have that autoantibody, But it's those patients that have the antibody that we think the statin most likely triggered it, and where we would avoid the further use of statins. And that would probably include statins that are found in dietary supplements, red yeast rice, and things like that. So, so getting tested for the antibody may be helpful. And another thing I would add is, I think Tom has seen some patients who have overlap of inclusion body myositis with statin-triggered autoimmune muscle disease who have that antibody. So overlap is rare, very rare, but possible.
I recently attended a seminar about using your own adipose tissue stem cells for infusions to treat autoimmune diseases, a variety of autoimmune diseases. And I'm wondering if that could be a type of treatment that could be beneficial for patients with myositis.
Dr. Lloyd, do you think you can take that one? I'm sorry.
You're giving me all the tough ones.
I know it's a favorite of yours.
Actually, last year at this meeting, I gave a session on actually what we call truth in advertising, evaluating claims, right? So obviously, stem cells are an extremely exciting area of research, and there are clinics now in the U.S. that are able to actually suck out fat and then somehow isolate cells and then inject them into tissues, so into muscles. Now, I'm not sure how these clinics are able to actually get away with doing this, given, quite frankly, there are no studies that actually support being able to do this. Really, as far as I know, in any disease, but certainly not in myositis. So I think the old adverb or proverb rather, actually buyer beware, right? So if you're seeing something advertised, whether it's a supplement or a stem cell treatment, if it's if there aren't any clinical trials or any clinical studies that have been done in this treatment, I would say just because someone is able to offer it and make a claim that it's helpful in any condition, really doesn't mean that it is worth your money really to try it. So in the case of injecting stem cells derived from fat, as far as I know, there's really no even plausible science out there that might suggest that your stem cells injected into muscle may actually lead to improved growth of muscle. So there's no animal studies, for example. And as we said earlier, I mean, there are always risks, right? So there are now reports of stem cells being injected into the brain and spinal cord that are leading to tumors. So I would certainly caution everyone against actually signing up for a treatment outside of a clinical trial.
I probably want to just add to that clinical trials, when you participate in a clinical trial, it's free for all patients. And so if you are seeing an advertisement that is an experimental therapy, but they're charging you thousands of dollars, you certainly want to be wary of that.
And I just add a quick
comment. So there are different kinds of stem cell transplants. The ones that you hear people with cancer, lymphoma getting are hematopoietic stem cell transplants. The stem cells that come from fat are short-lived mesenchymal stem cells, and there is no data that I know of to support the efficacy.
So FDA has actually started to look at it very critically, and they have shut down a number of these clinics, and a number of physicians who were doing these practices have lost their license. But still, I mean, it's a huge problem, and especially from California, where Dr. Goyal and I and Dr. Shea come from, it's almost an epidemic proportion there, where physicians are opening these so-called wellness clinics, and all they're doing is taking advantage of the desperation that some of these patients have. We see this in ALS, we see this in IBM, we see this in, I think, and some of more respectable hospitals are also getting into the act, which is really disturbing.
So I think we have time for one more question, and we'll go on over to this side.
My name is Marie Dominich. I was diagnosed 2 years ago with NM. Very fortunate the treatment has worked, but one thing that I do deal with is pain, chronic pain on a daily basis, which I've been told over and over again is nothing to do with the myositis, but is fibromyalgia. So I was wondering if any of you on the panel would agree that the pain is actually associated with myositis.
Yeah, I mean, so one of the problems is that pain in myositis is really not well studied. So there are two notions. One is that myositis technically doesn't cause pain or shouldn't cause pain. So why? So if the patient is complaining of the pain, it's likely due to fibromyalgia or some other associated feature. However, more and more studies are showing actually a lot of our myositis patients, initially when they present, they may or may not have pain, but even after they've actually gotten better, like you, their main complaint that is left is pain, because their weakness is improved, but the pain has now become a major component. And we are certainly looking into it. Is it an overlap with fibromyalgia? Because a lot of autoimmune diseases, like myositis is an autoimmune disease, has an overlap with fibromyalgia, or is actually something mechanically wrong with the muscle fibers and nerve fibers that are supplying the muscle that are causing the pain. I don't think it's clear yet, but definitely necrotizing myopathy is one of the areas where we see a lot of muscle pain, myalgia as one of the clinical feature of it. So there's definitely a link, but I don't think we are clear. Is it all the patients have a link with myositis as a disease, or is it overlap in many cases?
The other thing I would add is with necrotizing myositis, like Rohit said, pain is a very common problem, especially at the outset. Usually when people have muscle pain due to the myositis with the necrotizing myositis, they also have elevated CK levels or muscle enzyme levels. And usually in people who are being treated and who are well treated, the muscle pain goes away as the patients get stronger and the CK levels go down. Often the very first thing that we see when patients begin to flare, if we were like tapering their medication and then the disease sort of comes back, muscle pain is one of the first things people complain of. So I think one of the questions to answer is, do you have muscle pain because the disease is still active? And that could be addressed by looking at the CK or a muscle MRI or something like that. But there could be other reasons you have pain as well, even if the disease is not, the myositis isn't active.
I think it's a great question because it gives me the opportunity to tell you about OMORECT, which takes which is focused on patient-reported outcomes. It is not always the case that physicians who treat patients with myositis and patient with myositis consider the same features to be very important. And what we found is that patients regard pain, but also fatigue, in addition to the muscle symptoms, to be of utmost importance to be included into clinical trials, to take that into consideration. So I'd like to add that to everything which has been said. We have ignored it for a long time, but it's going to be a focus of us in the near future.
So that concludes our question and answer session, and I'd like to just thank our panelists for being here and for being so willing to talk to us. And so we can move on to our next session, which starts at 11 o'clock. Thanks, everybody.